Yeah. Good morning, everyone. Thanks for being here to the Fralin Biomedical Research Institute's Pioneers in Biomedical Research. Just a reminder, when the next PBR lecture is going to be November 9th by Dr. Jackson, talking about the blood tumor barrier. Today's speaker, I'm pleased to introduce, is Charlotte Boettiger. Dr. Boettiger is Professor of Psychology and Neuroscience at the Bowles Center for Alcohol Studies, Director of the Behavioral and Integrative Neuroscience Program at UNC Chapel Hill. She received her bachelor's in integrative biology from the University of California, Berkeley, her PhD in neuroscience from the University of California, San Francisco. She's an author or co-author of 50 articles and/or chapters. She's presented numerous invited lectures. She's currently a PI or co-PI on 2 NIAAA awards. Her honors include the Capillary Frontiers of Science Fellow from the U.S. National Academy of Science, and from UNC received a Research Mentor Award. For those who may not know, the Bowles Center for Alcohol Studies at UNC focuses on 3 areas of cognition: reinforcement-based association learning, selection bias for immediate rewards, and abnormal attention to addiction-related information. Dr. Boettiger and her team investigates how we form and break habits by studying how the brain enables us to learn and replace stimulus-response relationships. Please welcome me, join me in welcoming Charlotte Boettiger. Thank you.
Thank you so much. Thank you for that nice introduction, Warren, and thank you for the invitation to be here. I'm very flattered to be considered a pioneer. In the pioneer spirit, what I'm going to tell you about today is mostly work that's not published, so things that are on the verge of getting out into the world, and then some things that are in a newer stage, and then things to tempt you about what's coming in the future. With that, let's go. So I want to talk about where my work falls. Generally, if we think about problems like addiction, our ultimate motivation is to find out things that might help treat these disorders better than we're doing now. So some of you may know that we currently have 3 approved medications for treating alcoholism, for example. That's the same number when I started in this field about 20 years ago. So that's super depressing. They are as effective as antidepressants are for treating depression. They're underused, but we still could use more things out in this land here of FDA approval. So we're all familiar with what basic research is with, animal models and laboratory methods. And we all know what clinical trials are, where we try something out. There's several phases to it. And most things fail on this pipeline. And the failure rate is even higher for addiction related treatments than for other things, for reasons that aren't entirely clear. And the work that my lab does, I think of as falling in this kind of liminal space where we're kind of treating humans as basic laboratory models and trying to understand things about the neural mechanisms that can feed back into basic research, and we hope ultimately improve this pipeline. And not just for medication, so I've got pills here, but for other kinds of therapeutic interventions as well. And in particular, the approach that we have taken is trying to understand the neurobiological mechanisms of intermediate phenotypes. So intermediate phenotypes are quantifiable markers of some kind that differ in a particular population that you're interested in, and often are heritable. So people with a family history of that condition will fall somewhere between the control population and the affected population. And so in our case, we're interested in addiction-related intermediate phenotypes, and my training is in neuroscience. So we are exploring neurocognitive intermediate phenotypes in my lab. Why do we want to do that? Like other mental health disorders, addictive disorders are complex. They're heterogeneous. Anybody who, is familiar with Dr. Pickle's work has hopefully been familiarized with this idea. And we think that's one reason why a lot of treatments don't work very well. It's because their patients are not all the same. So we've got these various subtraits. You can think of intermediate phenotypes potentially as subtraits. And they're not just isolated, though. They interact with one another in complicated ways. And so we can have one patient that has, this in this constellation of subtraits, some are emphasized relative to others, and another patient looks quite different. So you might expect that the same treatment isn't going to work for both of these people. So what we're trying to do is basically isolate particular types of subtraits or intermediate phenotypes, try to understand those, and hope that that somehow can lead to better intervention strategies by people who are clinicians, not me. I'm a neuroscientist. And so that's kind of depicted here. If we can somehow understand these intermediate phenotypes, the neurobiology of those, the biology of those, that might help. That's all kind of set up. And as Warren kind of alluded to, my lab has explored intermediate phenotypes kind of along this continuum from abnormalities in stimulus processing, mostly attentional bias types of tasks. So attentional bias towards addiction-related cues, and more recently, attentional bias towards reward condition cues. We've also done a fair amount of work looking at decision-making behavior, so choosing smaller immediate rewards versus larger delayed rewards. I'm not going to talk about any of this work today. I'm going to very briefly talk about some attentional bias work if I have time. But mainly I'm going to talk today about abnormalities in this associative learning or stimulus response associative learning process. And these all are sort of under the domain of this perception action cycle that Joaquin Fuster put forward for describing the role that the prefrontal cortex is playing and perceiving what's happening out in the world and deciding how to use that to generate actions in a constant feedback loop. And in particular, the work that I'm going to talk mainly about is probing behavioral flexibility in people with a history of substance misuse. And why am I doing that? If we conceptualize the process of addiction, so you can't get addicted to something without trying it, right? So there's a fair amount of interest in trying to figure out why do people try things. But then there's also the transition from trying it, using it in a non-compulsive way into compulsive use. So a small proportion of people who initiate use of a substance will transition into this harmful compulsive use state. And many people who are in this compulsive use state achieve abstinence, but the big problem or one of the big unsolved problems is why people relapse. Drugs out of their system, they're out of withdrawal. Why do they go back and do this thing that they know is bad for them and is harming their life in most cases? And so one sort of foundation for what we're doing is the idea that perhaps deficits in behavioral flexibility might facilitate this transition. I don't think it's the whole answer, but I think that it could contribute to this, where we do the habitual action instead of something new that would take us in a different behavioral direction. And the idea is that this perception action cycle is somehow disrupted so that this transition back is facilitated. And this, we know, this ability to flexibly respond to the environment depends on the prefrontal cortex. So if you lesion the prefrontal cortex, we're not good at making a new response to a familiar stimulus. This is a kind of foundational review back from 2001 by Miller and Cohen, where they use this example where these are the cues from your environment. In this particular cue here is the telephone ringing, which normally triggers the response of answering the telephone. I realize this is a terrible example now because many of the younger people in this room are not familiar with this situation. You wouldn't automatically go to answer a phone, but in any case. But in this second context, you're in your boss's office and the phone rings. You do not answer the phone. If your boss is out of the office, you might step out and let someone know the phone is ringing. You might just sit quietly and try to ignore it. You might send a text to your boss, letting them know their phone is ringing. You're going to do something different instead of that. You're going to do something different instead of this overtrained automatic action. And being able to do that, to flexibly make a different response, depends on this intact functioning prefrontal cortex. And this fundamentally kind of looks like the addiction problem, right? We're not able to, when exposed to these stimuli, the context associated with our addiction, not able to do something different. And so that's kind of what we set out to try to investigate. And there's this idea that the prefrontal cortex represents the rules, essentially, that we use for how to respond to our environment. Just a picture. So when I say prefrontal cortex, I'm talking about this blue zone, this light blue zone here, which you can see is giant in humans proportionally. That's one of the reasons why I left basic neuroscience doing slice electrophysiology to come and do work in human subjects, because they have an enormous prefrontal cortex, and being trained as a neuroethologist, you want to pick your prep that has the most elaborated version of what you're interested in. So people interested in sound localization might study bats or owls, and I study humans. We also have known for quite a long time that addiction is associated with executive dysfunction. So probably anyone who does addiction work in this room has seen this diagram from Koob and Bolko. And one of the key areas that they point out is executive function deficits. So I'm going to try to probe a particular attention or executive function deficit with the work I'm going to tell you about today. So relapse, we can conceptualize it extremely simple-mindedly with the idea that you're exposed to a familiar stimulus, like someone offering you a cigarette, and your learned response is to take that cigarette and smoke it. But maybe you learn that that's going to give you cancer or lead to COPD, early death, all these things. And you decide then to develop a new response and say, no, thank you, I'm going to pass today. I'm not smoking now anymore. And to try to study that in the lab is tricky. And so we developed a task where people are exposed to these abstract visual stimuli, and they learn through trial and error to press a button that is associated with that visual stimulus. So a stimulus response learning task. We call it the habit because there's a hidden association between sets of stimuli that they intuit in the course of learning by trial and error. The task is rather fast, so they don't have a lot of time to think about things. They just need to respond, and they're rewarded for high performance, so they don't give up, essentially. So this is the first part, a training session where they develop extremely high accuracy in the task. And then we have them go home, at least get one night's sleep, It varies how long it is before they come in for their test session. But then the test session looks like this, where there's a initial part of the task where they're again exposed to those same stimulus sets that they learned. And then they also are introduced to new sets that they learn on the fly during the test session. So we can measure how well they are at executing those learned rules and how well they do at learning new rules. And when we compare people who have a history of a substance use disorder, but who are in recovery, in this particular study, I think the average was about 2 years clean and sober, but it varied a bit. And when we look at their accuracy in performing this task in the familiar sets, so the ones that they've practiced a lot before, that they were trained on in the earlier session in red, and the ones that they are newly learning during the session in green, you see that the people with substance use disorders are not impaired. They're as good, if not maybe slightly better at doing this task. And that in the course of this learning period, their performance on the new sets is almost as high as the ones that they had learned before. So then the key thing is we then give them a bit of a pause and we say, guess what? Some of the stimuli have a new correct response, and you have to learn that one now. And it'll be intermixed with sets that are the same. And we're going to still reward you for accurate performance. So they're still working pretty hard, motivated. And this response devaluation is going to give us a chance to then measure how many perseverative errors they make. So that everybody's going to make some errors as they search to find the new correct response, but we're interested in the proportion of perseverative errors, making that old correct response. And at this point, they've typically had something like 900 trials. So it's very highly overtrained responses that they have learned. And what we find is that unlike the learning the sets or executing the familiar sets, the people with a history of a substance use disorder are impaired relative to the control subjects. They make more perseverative errors. And that's true only for those familiar sets. The novel sets, even though they're performing at close to the same level of accuracy, they can quite easily switch to a new correct response in that case. And so we think this is an index of having generated an automatic or habitual action selection strategy. Like they're able to switch and do that more quickly. And one thing I like to mention when I talk about habit is that's not always a bad thing, right? Being able to automatically respond to a stimulus is important for music performance. It's important for sports performance. It's important for firefighters. It's important for soldiers. There's a lot of conditions under which not thinking things over and just acting is very important. And in all of those cases, people do massive amounts of training, doing the same thing over and over and over and over and over, so that when you need to do it, you do it. And it can be adaptive, right? So it's not necessarily a trait we would want to eliminate. And it may be a trait that predates the substance use. We don't know about that, although there is some data from Kathy Grant's lab and monkeys that shows that animals who have a tendency to have inflexible behavioral responses before alcohol exposure tend to be the animals that acquire compulsive alcohol self-administration more readily. So we'll see. Yeah. Mm-hmm.
So that being said, do you do anything to ensure that your controls don't have any of those, like the propensity to make those decisions like that? Like, I don't know, I'm just thinking maybe obesity or something else where people are making habitual decisions against their health, regardless of substance abuse disorder.
That's a good question. And most of our studies, we don't collect information about weight or height, although in some we do. But that's a good question. I think that probably, those things probably could be contributing to error within the group. Mhm.
There's always the issue of executive function versus attention.
Yep.
Because, you could as easily explain this as an intentional error.
Right.
So how do you control for that?
That's a good question. And you're right, it could be attention and not working memory. We know that in order to execute or to learn these stimuli, you need dorsolateral prefrontal cortex to be engaged. And it looks like synchrony between the supplementary motor area and the dorsolateral prefrontal cortex is important for learning these, and that the orbital frontal cortex gets recruited in this condition where we're trying to suppress the old response.
Is there anything in the acquisition phase that you could further grow to distinguish between the?
Yeah, so we do have a little bit of data that suggests that activity in the putamen during the initial learning phase, higher putamen activation in the learning phase predicts more habitual responses in the test phase.
Given that putamen has unknown connections still.
Yeah.
It's interesting.
Mhm. Yeah, there's a lot of, I think there's a lot of questions being generated by what we've found so far. There's a,
Separating.
it is, there's a window that's come up here instead of my. There we go. So the next thing that we decided to do using this task was to ask whether we could shift people's action selection using a non-invasive tool. So we use transcranial alternating current stimulation. We apply these carbon rubber electrodes to the scalp. We did it dorsolaterally over the, bilaterally over the dorsolateral prefrontal cortex, applying 10 hertz stimulation, which is the average alpha peak for most humans. A community sample of adults. And just as a reminder for folks, if we record electrical signals from the scalp using EEG, for example, we can see activity in particular frequency bands. So the lowest frequency band or the slowest oscillations are the delta, and then there's the theta alpha, which is quite large. I'll show you some pictures that show you how large and noticeable the alpha peak is in most people. And then there's beta and gamma, and the height or peak to peak difference in these oscillating bands is known as power. So it's like how much power is there in these different frequencies. And previous work has shown that if you measure power in the alpha band before stimulation, and then apply alpha stimulation, you see that the power in the alpha band goes up. And this is the stimulation period here in gray. And when stimulation shuts off, you see that alpha power is still elevated. Why did we choose alpha power? Part of it had to do with our collaborator, Flavio Froelich, who runs the Center for Neurostimulation at UNC. And they had done some work showing that alpha stimulation seemed to boost creativity. They used the Raven's matrices to measure before and after. So we decided to give that a try. In retrospect, I think I would have used a different frequency, but this is the data that we have.
Question?
Yep.
Is it a function of, you get entrainment to the stimulation frequency, increase the power? So if you do that experiment you showed on the previous slide,
yeah,
with the stimulation using different power windows and the different frequency ranges, can you bring virtually any component, delta, whatever, into that kind of entrainment?
That's a good question. I suspect the answer is probably yes, but I don't know for sure. It's a bit of a frontier, the TACS. Most of the work that's been published today has been TDCS, where you just put a direct step function of current. I suspect you can boost power with any of the frequencies, but there's less data for other frequencies.
The reason I asked is I was just curious if to what degree different components of the intrinsic, cortical circuitry are contributing to the different frequencies, and whether a simple synaptic modifiability processes such as potentiation are going on, that are the different components that are driving more one frequency or the other
Right.
sensitive to.
Right. Great question.
Okay.
Yeah, so I know the answer to that. So in this study, it was very similar to the earlier one I talked about. They come in for habit training and they come in for a habit test. In this case, they came in 2 different times, one in which they got sham stimulation. So there's stimulation on the ramp up and then we shut it off, and stimulation on the ramp down. It's an effective blinding method, so they can't guess which session was which, and the researcher who's doing the test is blind to the condition as well. So this is within subjects, so they come back and get the other stimulation, orders counterbalance. And we compared adults with an SUD history again versus controls. And we are happy to find that, just like in our earlier study, the people with SUDs, again shown in a dashed line, they're able to do this task. And there was no difference between the sessions where they were about to get real stimulation versus when they were about to get sham stimulation. So those are good controls. And this work is all led by my former grad student, Teresa McKim, who's now faculty at University of Nevada, Reno, and a former RA who's now out there in the field as a clinical social worker. And what we found was that bilateral TACS at 10 hertz to the dorsolateral prefrontal cortex, the effects depended on addiction history. So in control subjects, here's the sham condition in white and the true stimulation in gray. They got worse. Their performance was more habitual. And the people with a history of SUD, there's like a hint that it was going down, but not significant, but the interaction was significant. This is not what I wanted, right? I wanted to see something that would be doing nothing to these guys and making these guys better, but that's not what we got. That's why you do the experiment, and then you try to figure out what's going on with these results. And what we did was try to figure out what was going on within this particular sample, and if there was anything that there's a large amount of variability, and we wanted to know, is there anything that might explain that variability in that group? And what we found was the explanatory variable was the duration of their substance abuse. So if they had only been, if their duration of substance abuse had only been a couple of years, they looked like the control subjects, where they became more habitual in their action selection, just like the control subjects. But for those who had had a longer history of substance abuse, the stimulation actually seemed to improve their perseverative actions. And when we controlled for stimulus order and for age and for their performance in the sham condition, it was a pretty strong correlation. And then trying to figure out, well, what's causing this. One thought we had is that maybe when we were stimulating at 10 hertz in the control subjects, we were hitting their alpha peak, but maybe for the people with a history of substance use, there was some kind of systematic shift in their alpha frequency associated with their duration of substance use. Here we're looking at power on the y-axis, getting increasingly higher frequency, normal individual alpha frequency is found here, but maybe our control subjects were getting stimulated effectively at alpha frequency, but maybe the people with a long history of SUD were being stimulated at a different frequency. Their alpha frequency was shifted. That's just speculation. There wasn't any data in the literature that we could find. We didn't collect EEG, so we couldn't know. But another thought, which we have some evidence to support, but I won't talk too much about, is that maybe TACS is activating central catecholamine release. And why do I think that? Well, you've got cranial nerves innervating the dura and the scalp, and the mostly the trigeminal nerve. And the trigeminal nerve sends projections back to the locus ceruleus. And so maybe all the effects that we're seeing are due to locus ceruleus activation, releasing catecholamines up here. And we know from a lot of work from Amy Arnston and others that there's an optimal range for prefrontal function, and that either too much or too little can be impairing. So maybe our folks with a history of substance use disorder are over on this end and they were helped, and maybe the control subjects or short duration folks were overdosed with catecholamines. Warren?
So in pharmacology, we often study rate dependent effects. Baseline determines the effect of the intervention.
Mhm.
It's been documented with various pharmacological agents and behavioral interventions. Have you considered doing a rate dependent analysis here where the control subjects who start with a higher baseline go down and the ones with a lower baseline go up?
Mhm. That's a good question. So in the people with a substance abuse history, we did control for their baseline or sham performance. So I don't think that that was it, but that's a good question. And a final thought, and this was influenced by my collaborators and colleagues in the Alcohol Center, is maybe the effects were due to residual effects of adolescent alcohol misuse. So my colleagues who work in animal models have shown in a variety of different ways that the adolescent brain is uniquely vulnerable to alcohol insult, and particularly there are long-term effects in the prefrontal cortex and in behavioral flexibility that last into adulthood. And so maybe these long duration folks had some adolescent alcohol misuse that was leading to these issues. We do have some acute stress data that supports this idea to some extent, but I'm not going to talk about that today. So what we decided to do is another study where we would measure people's individual alpha frequency before the stimulation. So they come in and get these high density electrodes placed, and then we record the resting state EEG signal. And this is the electrodes from the prefrontal area. You can see this nice alpha peak. It's pretty stable across time. So this same person came back weeks later, and you can see the same alpha peak in about the same spot. And so in this study, instead of giving everybody 10 hertz stimulation, we're going to find their individual alpha frequency first, and then we're going to stimulate them at that individual alpha frequency. And I was able to do this thanks in part to my collaborator, Dr. Margaret Sheridan, a clinical psychologist who co-mentored my graduate student, Maddie Robertson, and is an EEG expert. So brought me into the EEG domain. And I should mention that Dr. Danita Robinson is my collaborator on all these projects, and she's doing sort of parallel work in animal models and rat models to try to figure out sort of related and complementary things. Oops. Now, as we were getting this study started, I went to this Kavli symposium where it was like science camp kind of, where people were presenting all kinds of exciting, fun things. And I ran into an old friend who was doing some work looking at a particular feature of the EEG recording where you could map out the power as a function of frequency. And you see these big oscillatory peaks, especially the alpha peak here, but you can subtract off that stuff and you can measure the slope of the aperiodic or noise signal in the EEG. And what he found was that there was a difference when you compared that slope of younger adults relative to older adults. So older adults had shallower slopes. And it was meaningful because if you plot the slope versus their accuracy in a working memory task, you see that the older adults are up here, they're less accurate on average, and the younger adults on average are more accurate, and that there's a significant correlation between their slope and their accuracy. And so if you do a statistical model, you can see that you can account for the effects of age on accuracy largely through this slope measure, this 1/f for frequency measure. They call it noise in this initial paper, and they've moved away from that term. So I saw this and I thought, we're getting EEG data in the work that we're doing. And not only is this sort of interesting in terms of something that might regulate a prefrontal dependent performance, but there was a lot of data that came out. I guess it's the next slide, sorry. So we're going to measure slope in this aperiodic slope in our data. And there was data that came out where they're using recordings in animals to show that this slope, which you can also measure in local field potentials, is related to the balance between excitation and inhibition in the region where you're recording. So that a flatter slope is associated with a relatively higher excitation. Now that can be accomplished different ways. It can be that inhibition is suppressed, or it could be that excitation is elevated, or it could be a combination of both. But relatively speaking, a flatter slope is associated with a more excitable state than a steeper slope where you have a dominance of inhibitory stuff versus Mike.
So it reminds me to ask you, I meant to ask you in a previous slide, but both for the power spectrum for an individual, their particular signature of power and alpha, and for the slope metric as well, how stable are those measurements in a given individual? I guess I'm trying to understand over what time frame you need to collect the data. If you do it 30 minutes later, an hour later, are they unique signatures that?
It seems to be fairly stable, but I'm gonna I've bet the farm on it being something that we can manipulate.
So over what time period do you collect the data to establish a curve like that?
It will be a very short period that we're planning to do. So in this particular case, we only collected this data before we did our simulation, and we didn't collect EEG again afterwards. My whole scientific career, I feel like, is built on this process of like, why didn't I get that data? And so then the next study, we get that data, and then there's some other data that I wish we had gotten. Climbing a mountain or something. Yeah, it seems to be a relatively stable trait, but I think it's manipulable. All right. So I'm going to get back to this idea about adolescent alcohol exposure, and I want to put us all in the mindset of thinking about how the prefrontal cortex develops. So here's a model from a work from Tom Insel, where if we look at age from 0 to 25-ish, and the percentage of maximum, looking at prefrontal excitatory synapses versus inhibitory synapses, can see that in the early years of childhood, it's very excitatory dominant, right? There's not as many inhibitory synapses until we hit kind of mid-teens, and then there's a big ramp up in inhibitory synapses, and the ordinary course is that the inhibitory synapses dominate in the prefrontal cortex. And so there's this shift between dominance of excitation versus dominance of inhibition. And we're interested in what's happening if we're dumping this indirect GABA agonist, alcohol, into this developing system. Maybe this is one of the reasons why the adolescent brain is more sensitive than the adult brain, because this dynamic process is happening here. And we hypothesize that one of the effects of this exposure is that somehow this ratio ends up being different in the adult brain. We're going to try and investigate that. Can we detect differences in this EI balance in adulthood associated with adolescent alcohol use? So we're going to be looking at that slope measure. And indeed, we do find that. So if we plot the slope here in our control subjects versus people with a history of adolescent binge drinking, see a significantly shallower slope. Average the fits together, they look something like this. And this is controlling for their age, their sex, their current amount of drinking or current alcohol misuse, and for lifetime drug use. And in this study, we excluded anyone with a lifetime substance use disorder. So this is encouraging to us, but then even more interesting to me is that if we plot this slope value, we see that more habitual behavior in our habit path is associated with a shallower slope. So sort of what we might have predicted as the shallower slope in the prefrontal cortex. We know from that earlier data I showed that working memory is impaired, and here we find that perseverative error is increased in our tasks. And if we run a model where we take the adolescent binge score and the habit perseverative errors and the EEG slope, we find that there is a significant mediation of this adolescent alcohol effect on perseverative error mediated by EEG slope. And might be asking why there's two different numbers there. The case where we see the significant effect is only in our participants who had their sham stimulation session first. If the one of the other stimulation conditions intervened before we measured the perseverative error in their sham session, this relationship didn't hold, which suggests that the stimulation is doing something to the excitation that ends up interfering with this relationship. But we're pretty clear from our data that it looks like adolescent binge drinking decreases the slope, so shifts the balance towards excitation from inhibition, and that's associated with increased perseverative error. And along the same lines, as we were getting the study started, there was a parallel arm in which we were doing fMRI. And I'm not really talk about any of the fMRI results today. That's sort of to come. But I read a paper from an old friend using MR spectroscopy to look at GABA and glutamate levels in the prefrontal cortex. He was looking at people with schizophrenia. And I thought, well, that would be a nice complement to this work that we're doing, something a little closer to getting down to synapses, right, than this sort of somewhat abstract EEG measure. So another excellent grad student in my lab, Elena Vidrascu, placed these voxels over the prefrontal cortex in our participants. We can parcellate that into gray matter, white matter, CSF. And when we average across subjects, you can see that the placement was quite reliable across people. And we get these nice traces where we can see the magnitude of glutamate and glutamine here and GABA here. And in this plot, you're seeing people with a history of adolescent binge drinking in blue and control subjects in green. You can see that the adolescent binge drinkers had higher levels. Now these are adults, right? So this is based on their self-reported adolescent activity, but higher levels in both measures. But what we're really interested in was the ratio of these two substances. And, oh,
Stand exactly like
okay.
So is this structural MRI or is this
No,
MRI?
it's MRS, it's spectroscopy.
Okay.
So we place a voxel over this area, and instead of in normal MRI, structural MRI, functional MRI, we're looking at water signals, the peak of the water signal. And in this case, we adjust the sequence so that what we're looking at is these molecules, the glutamate and glutamine. They're mixed together. We can't really disambiguate them, and GABA. So we can't say it's not synaptic necessarily. It's just in there.
It's what's
Right?
there.
It could be outside the synapse. It could be inside the synapse. It's kind of gross, but it goes in a little deeper than the EEG measure. And what we find is that this ratio predicts perseverative errors. So in the heightened inhibition, so what we might think of as the healthy adult state, or more towards the healthy adult state, we have fewer perseverative errors, and with a with a more glutamate, glutamine dominant spectroscopy measure, we see more perseverative errors. So this leads us to feel increasingly confident that adolescent alcohol exposure is perhaps shifting, and it could be this direction as well, but it's shifting that balance between excitation and inhibition. I realize I think I left out a slide. We find, no, it's coming, never mind. So remember, this was all a lead up to doing another TACS study, and we found that we replicated our results. So giving people stimulation at their specific individual alpha frequency didn't make any difference. We still saw that the control subjects increased their habitual responding with alpha stimulation, and that the people with a history of adolescent binge drinking, somewhat like the people with a history of substance use disorders, there was a slight trend to go down, but not a significant effect, even though we had individualized their alpha. And I didn't talk about this yet, but there was a third session for this study where we gave theta stimulation, and we found that it didn't do anything for anyone, but it helps us feel confident that the alpha effects are frequency specific. It's not just having your brain stimulated that's doing this. And we found that response to that alpha stimulation is related to an excitatory inhibitory balance. So this slope measure that we acquired is associated with how we responded, how our participants responded to the alpha stimulation. So if they had a shallower slope, they tended to reduce somewhat their habitual responding, and steeper slopes, like the control subjects, tended to have showed more habitual responding. I'm looking at the time, and I think that I want to leave time for questions. So I'm going to not talk about our attentional bias results, except briefly maybe to say that perseverative errors are associated with increased facilitated attention to reward condition cues, which is not what I expected, but it gets at your point that are we really measuring something going on with attention here? We may be. And in our next study, we're going to collect EEG and spectroscopy before the stimulation and again after the stimulation. And we'll be doing this on a 7T scanner, so higher resolution image. And I'm going to skip over that. These are what some of my other graduate students are doing. And I just want to thank my lovely team for doing all the work, aside from the grant writing, and I'm happy to take any questions that folks have.
You compared controls and people with substance use disorder. Have you compared people with active substance use disorder versus those in recovery?
We have not. I think partly, it's difficult. Their lives tend to be a little more disorganized. It's harder to get them in. I think if I had access to a clinical group who had patients, that might be easier, but we don't have that population.
So I'm a little curious about the relationship between the overall synapse addition and elimination, which, as you know, the only really reliable data was generated in rhesus monkeys. And I was there when it was done. And I think the idea of who are the inhibitory synapses and who are the excitatory synapses during the period of elimination is not clear. And there's a lot of evidence for functional elimination of inhibition as well as excitation. So I'm wondering how you think about the output in terms of the underlying synaptic architecture. Is it that there's a selective change in excitatory input synapses, local excitatory synapses, or subsets of inhibitory synapses?
Great question. So there's a tiny bit of data that suggests adolescent alcohol exposure in rats causes selective loss of parvalbumin positive inhibitory neurons in prefrontal cortex, which they've been implicated in trying to sort of synchronize prefrontal output or cortical output. And so that's one thought, is that perhaps there's sort of a disorganization of prefrontal synchrony caused by that adolescent alcohol exposure, maybe from GABAergic cell loss, and that makes the prefrontal cortex less efficient at sending its messages out.
Yeah, I mean, there have been these studies from the longitudinal changes of gray matter and white matter. I mean, they're not absolute, but if you looked at the gray matter volumes in your early alcohol exposure group or something like that, do you think you could pick something up?
That's a good question. We probably could, actually.
Just as a, as a look and see, because you'd have the data.
Yeah. One of the things we're doing, one of my students is very interested in aging. I can't move it. Where we've taken the structural images from this most recent study and fed them into a program called BrainAger, which allows us to get basically differences from predicted brain age. So they have a model based on like 3,500 brains, where they can predict the age of the brain that you put into it. And then you can look at the difference between their chronological age and their predicted age. And there's data in the literature showing that people who drink a lot, their brains are aged a bit more than people who don't. And our data suggests that adolescent drinking, independent of your current drinking, also is associated with increased brain age. And so we're trying to replicate that in some other data sets.
And just quickly, do you ever look at interhemispheric synchrony? Or,
We have not.
but
Yeah, I mean, we have.
in terms of all of the, the accounting of the who's connected to who,
Right.
that would actually be an interesting thing to consider.
Maybe follow on that a little bit, this mechanistic question. First, really simple one. Is it known that an early alcohol exposure lowers seizure induction thresholds in terms of potential damage to local inhibitory circuitry?
Scott Schwartz lab, you probably know that. They've done slice physiology to look at the.
And then secondly, much more mechanistic, and I realize you're not likely to have the answer. I'm just curious what you think. So this idea of putting on a GABA, this GABA agonist that leads to either a destruction or a decrease in efficacy or whatever it might be on the GABAergic neuron synaptic outputs, is idea of what that mechanism is of the agonist leading to the probably retrograde damage to the synapses. Is there a hypothesis about that?
My collaborator, Danita Robinson, is doing some work. I think it might have to do with an increase in the perineuronal nets surrounding the parvalbumin neurons. They're trying to freeze up the plasticity there. There's also a group from Fulton Cruz's team that has shown that it seems to be mediated, adolescent alcohol effects on the brain seem to be mediated by inflammation. And so they're doing some work showing it's a good news for people who drank too much when they're teenagers that exercise and anti-inflammatory treatment, they use Ibugilast, seems to reverse some of the effects of adolescent alcohol exposure in rodents. That might be something that contributes to the variance.
I guess kind of along those lines, I was wondering if in the correlations that you did with, I guess it was alpha frequency against the substance use disorder duration, if you had looked at sobriety duration, because you said that kind of varied in your.
We looked at that and that didn't seem to be a factor. So that leads us to think that either it's some kind of long-term persistent thing, or was it a pre-existing thing, or is it both?
Right.
Both.
Okay.
Very good. Thank you so much.